Hello students, myself Harish, Biology faculty from Global ITs. In this video, we're going to have a look on some of the important questions which may appear in our coming examination. So now let's have a look onto the question. How many autosomes does a human primary spermatocytes have? So they're just asking here, what is the number of autosomes in the human primary spermatocyte? The question appears to be easy. It looks easy, but here we make a silly mistakes. So now, first of all, we have to know what is autosomes. Guys, remember the chromosomes, we broadly classified them into two categories. One is called as a autosomes, whereas the other one is called as a sex chromosomes. Okay, so now we got to know what is actually the autosomes. Now we look into the human primary spermatocyte. See, the human primary spermatocyte is actually the result of mitosis seen from the spermatogonium. So the spermatogonium undergo the mitotic division. As a result of mitotic division, it results in the primary spermatocyte. Now here, what will be the ploidy maintained? What will be the chromosomal number maintained? That is important. Now, hence it's undergoing mitosis cell division. Mitosis cell division. Of course, it's going to be the equational cell division. What do the mother cell have? The same number of chromosomes are seen in the daughter cells also. Means this primary spermatocyte is obtaining from the spermatogonium. Okay, the spermatogonium have a diploid set of chromosomes, and definitely the primary spermatocyte also will have a diploid set of chromosomes. Okay, now one point is clear. Now look into the options here. 46, 23, 44, 22. That's the option given. So here we have to be very much careful. They ask only autosomes. Okay. Now let us say the mother cell having a 46 number of chromosomes. Definitely the daughter cells will also get the 46 number of chromosomes when they are undergoing mitosis. So let this be our spermatogonium and this will be our primary spermatocyte. So this is a chromosomal number. Now what they ask? Autosome. So autosome is going to be, listen, in human case, if we have 46 number of chromosomes, among this 46, 44 are autosomes, whereas the two going to be the sex chromosome. So in mother cell they have 44. Definitely in the daughter cell also it's going to remain the same. It's going to be 44. In spermatogonium also we see the 44 autosomes. In primary spermatocyte also we see the same number, that is 44 number of autosomes. So that the correct option going to be B, 44. Guys, 46 is given, which is pretty much confusing here. 23, because we always say 23 page, this is also pretty much confusing. And remember, if they ask you for the secondary spermatocyte, then only it's going to be a 22. So all are similar options, but the correct option going to be 44. In autosomes of primary spermatocyte, we see 44 number of chromosomes. So that's all about this question. Now we'll move on for next question. The process of formation of haploid motile male gamete from a haploid non-motile spermatids are called as dash. That's the question. Okay. Now we'll see what are the options they are given. The first one is spermiation, oogenesis, spermatogenesis, spermiogenesis. Okay. So now in general when we have a look into it, it appears to be what is the formation of sperms are called. That's it. Okay. When such things strikes our mind, we have a chances of marking spermatogenesis. But here they ask this clearly: haploid non-motile male gamete from a haploid non-motile spermatid. So we should be cautious here and we know what is this, what's actually asking. See, listen students, here the spermatogonium undergo mitosis cell division. As a result of it, we are going to get primary spermatocyte. Here the mitosis is equational division, like we saw in earlier previous question. The same number of chromosomes is maintained here. Now this primary spermatocyte undergo a reductional division called as a meiosis. As a result of it, we see the haploid secondary spermatocyte. So this is diploid. Their daughter cells are haploid, means the number of chromosomes are reduced here. Okay. Now, the secondary spermatocyte undergoes meiosis so too, where we can see the formation of non-motile haploid spermatid. Now, this spermatid will undergo certain changes and forms a haploid motile spermatozoa. Okay, now the haploid motile spermatozoa is forming from the spermatid, and this process of formation of spermatozoa from the spermatid are called as spermiogenesis. See here the terms appears to be same: spermiation, spermatogenesis, spermiogenesis. Okay, so we should be cautious here. We have to mark the correct answer, that is spermiogenesis. Then what is the spermiation? Spermiation is the release of matured spermatid from Sertoli cells to the seminiferous tubule. And oogenesis, you can easily eliminate this because that's a formation of ovum. And spermatogenesis, whatever we discussed here, right from spermatogonium till the formation of spermatozoa, the whole thing is spermatogenesis. The spermiogenesis is the correct answer, where the spermatid it's formed into spermatozoa. Now we'll move on for the next question. So if a cross made for the given characters, what will be the possible phenotypic ratio in the F2 generation? The given characters are dominant round allosates and recessive wrinkled allosates. Dear students, first we have to see what they are asking specifically. So what they are asking a possible phenotypic ratio in F2 generation. So we have to know the phenotypic ratio. So first we have to be aware what is phenotypic ratio or what is phenotype. Okay. Now we all know the physical appearance is called as a phenotype. Okay. Now how to identify this? Even though the question appears to be tricky, it's easy. See, first of all, have a look here. They mentioned dominant round allosates. So let us consider them as a parent. Now we are making a cross. Okay. The parents are dominant round, capital R, capital R. Let the genotype of the round going to be capital R, and genotype of allo going to be capital Y, capital Y. And now we have a recessive, that is recessive wrinkled and green cells. So recessive, we write it in the lowercase, small r and small r, and the green is written in the small y and small y. Okay, so small r, small y, small y. Now when they form the gametes, so capital R, capital Y, and here small r, small y. Now they form the F1, that is capital R, capital Y, small y. So this is going to be the F1. So F1 going to be all are heterozygous. So all are heterozygous round allo. All are heterozygous round allo. We know that when we see the both dominant and recessive, both the genes, we call them as heterozygous, or if they have any one kind, whether it may have only a dominant one or only recessive, then it's going to be called as a homozygous. In this case, we got heterozygous condition. Whatever we got in the F1, all are heterozygous round allo. Now we will see. They ask for which generation? They ask for F2 generation. So we'll move on for the F2. What is F2? F2 going to be selfing. That is F1 should be crossed with the F1. So in F1, what is the gene? That is capital R, capital small r, capital Y, small y. So this is one parent and heterozygous round allo. That is capital R, small r, capital Y, small y. Okay. Now we will see the gametes. Okay, so now the gametes going to be capital R, capital Y is one gamete, capital R, small y going to be the other gamete, and small r, capital going to be the third one, and the fourth one going to be, I'll write that over here. So small r, capital Y, and small r, small y going to be the another gamete. So hence it's same, we'll write the same here. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Done. Now we'll just draw the Punnett square to see the what will be the possible phenotype. Okay. So now the Punnett square will be like this. So here we'll write the gametes. Okay. So checkerboard, or we also call this as a Punnett square. Okay. So now we'll write the gametes of both the parent at each side. Okay. So now we'll take one parent at the top and one parent over here. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. One parent is done. Now we'll write the another parent over here. That is capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Then what we have to do right now? Just we have to keep writing whatever the I is present here. Okay. So capital R, capital R, capital Y going to be the result here. So here capital R, capital R, capital Y, small y. Okay. And the next thing, capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. So in the same way we have to do for each box here. Capital R, capital Y, small y, capital R, small y, capital R, small r, capital Y, small y, capital R, small y, small y. Okay. Now we'll see the next one. Capital R, small r, capital Y, capital R, small r, capital Y, small y, small r, capital Y, and here also small r, capital Y, small y. Okay, and the last one, capital R, small r, capital Y, small y, capital R, small r, small y, small y. Okay, and here small r, capital Y, small y, and at the last small r, small y, small y. Now we got to the products here. Now let us check what are those. Here we got round yellow. So we'll write at this corner how many round yellows are seen. We'll write it over here. Round yellow. So how many round yellow are visible here? Let us keep writing. One, another round yellow going to be here. Round yellow. Two, okay. Two. Here it also round yellow. Three, round yellow. Four. Okay. How we are identifying it? Very easy. See, the dominant one is expressing. Okay. So capital letter is represent the dominating character. So if it's a capital letter is present, so that we can say the expressing character is dominated over there. So that is expressing. That's going to be the phenotype. Okay. So fine. Next here also capital R is there, so it's going to be round. Capital Y is also there, so it's going to be yellow. So round yellow. So here also round yellow. Here we have a capital R, round, but we have a small y, small y. So what is a small y here? It's going to be the green seeds, right? So we'll take another one that is round green. Okay, round green. We got one. Round green, we got one. So let us mark like this. So now I'm giving the squares for the round yellow And I am giving a circles for the round green. So what is the next one? Round yellow. So one more round yellow we receive, and here round green. Okay, so round yellow, and this one going to be round green. We are left out with two rows. Round yellow, round yellow. Got it? So this is also round yellow, and this is also round yellow. Two more. One, two. Okay. Now fine. Next, what is small r, small r? Guys, it's a wrinkled. So we are getting one more character here. That is wrinkled yellow. Okay, we'll write wrinkled yellow. So we're getting one more. Okay. So this is a box, and this is also a box. And look at the next one. I'll give a different shape for this. Okay. So this is going to be the wrinkled yellow. How about this one? This is also wrinkled yellow. Have a look here. This is also wrinkled yellow. Fine. So we got one more wrinkled yellow here. At the last row right now, capital R, small y. Guys, round yellow. Make a mark. And here, capital R and small r there. So round, it's going to be round, and this one going to be a small y. What is this? The small y is representing green. So it's going to be a wrinkled green, right? So we are getting a, or is it wrinkled yellow? Sorry, it's going to be wrinkled yellow, right? It's going to be wrinkled yellow. So this is going to be wrinkled yellow, and previous one round green, right? Round green. So it's going to be round green, and the last one small r, it's going to be a what it is, small r? It's a wrinkled green, right? Wrinkled green. So only one. Let's have a count here. 5 plus 4, 9, 3, 3 and 1. So we are getting 9 round yellow, 3 round green and 3 wrinkled yellow and 1 wrinkled green phenotype. So now we got to know what is the phenotype. Now the ratio, if we write down, it's going to be 9 round yellow, 3 round green, 3 wrinkled yellow, and at last 1 wrinkled green. So this is the ratio they are asking. So which option have this ratio? 1 is to 2 is to no, this is not. 9 is to 2 is to 3 is to 4, no, 2, no, and the last one, option D, going to be the correct answer. That is 9 is to 3 is to 3 is to 1. Okay, so this is how we have to tackle this kind of a question. It's very simple. It's a simple dihybrid cross. They just asked us to just see what is the phenotypic ratio, which is going to be 9 is to 3 is to 3 is to 1. Now here is a next question. What will be the F1 generation where the red flowers of Mirabilis jalapa crosses with the white flower? So here we have to make a cross between the red flowers and the white flowers. Okay. Now guys, very important thing we all have to know here, the example they have given. That is flowers of Mirabilis jalapa. Okay. Let us underline that and just move on. Guys, we'll take a dominant red genotype as capital R, and let us have a genotype of white as small r because they are recessive in nature. Now they are going to form a gametes, capital R and small r respectively, and after that they are going to form a F1, whereas capital R and small r respectively. We got heterozygous condition. Now what is this heterozygous? Is it heterozygous red? Now can I call this as a heterozygous red? Okay, can I call this heterozygous red? Then the answer going to be no. So it won't be the heterozygous red. Why? Now I'll tell you. When you take the monohybrid cross, now let us say we are crossing a tall plant with a dwarf plant. Now here also same thing, we'll get the tall and the dwarf gametes, they fuse and they form a heterozygous tall. Here it is a heterozygous tall, right? So now it's almost the same, right? But very important key thing we have to understand, guys. Here the example they have given is Mirabilis jalapa's flower. We all know that Mirabilis jalapa's flower is a very good example for incomplete dominance, right? Now here what's happening? Both the alleles are partially involved in forming the heterozygous capital R and small r. So hence we get the intermediate character here, that is pink color. So that is why it won't be red. So we'll cancel out the red, or it won't be white. Then the correct answer going to be pink color. But see, that's why this is a monohybrid cross we see with respect to the Mendel cross, right? So that's why here there is a variation seen when compared to the Mendel crosses. So here it won't be the dominant one of their parent in the heterozygous condition. It will be the intermediate character between the parent. So it's going to be the pink is the correct answer. Now we're going to have a look into this incomplete dominance in detail. In this video, we're going to talk about a very important concept, incomplete dominance. Now let us understand what is incomplete dominance. Incomplete dominance is a phenomenon in which the F1 hybrid characters will be an intermediate of the parental gene. So very simple, whatever the F1 hybrid characters we are getting here, it will be intermediate that of the parental gene. So here, a very important thing we have to note, the phenotypic ratio deviates from a Mendel's monohybrid cross. So generally, whatever the Mendel's monohybrid ratio we saw, that is 3:1, it won't be same in this case of incomplete dominance. Okay, guys, it is quite commonly seen in the plants of 4 o'clock plant and snapdragon. Now, let us understand detailing with the illustration of incomplete dominance. Now, we are crossing a two-colored flower, that is red and white. Their genotypes are represented as capital R and small r respectively. Here, when the gametes are obtained, they undergo F1 generation. As a result of F1 generation, we are getting capital R and small r. That is, in a heterozygous condition, we are getting a capital R and small r. Now here, if this was a normal, a general Mendel cross, we used to tell it's a heterozygous red. But here, we cannot say that. Here, it's showing an intermediate character, that is pink. So pink is the intermediate between red and white. So now let us move on over here. For F2, that is selfing, F1 has to harmonize with F1 here. Here also, the genotype is capital R and small r. What we obtained is same here. When you just take the F2 here we get one homozygous red we are getting here, two heterozygous pink we are getting here, and one homozygous white flower we are obtaining. But if you remember monohybrid cross, there we used to get 3 a dominant character and 1 a recessive. But here we are getting 1 dominant, 1 recessive, but here the 2 intermediate flowers are same. So this is what a ratio deviation, that is a phenotypic ratio is deviating here. So this is also called as partial dominance. See here, the 2 alleles which is responsible for red and as well as the white, they are partially involved over here. So that's why we are calling them as a partial dominance. Enzyme of the pollen is dash. Hard outer layer of pollen grain made up of cellulose and pectin. It withstands high temperature. It cannot be degraded by any enzyme. Which one of the following statement is correct? The options given are option A, statement A and statement B. Option B, statement A, statement B and statement D. Option C, statement A, statement D and statement C. And option D, statement B and statement D. Dear students, here we have to check which among the statements are correct with respect to the enzyme of pollen grain. Now, to answer this kind of a question, we should be aware of concept, that is enzyme of pollen. Now, the pollen is made up of 2 layers, inner layer and as well as outer layer. Okay, so the outer layer we call it as a enzyme, whereas the inner layer we call it as a enzyme. So what am I writing now? It's a enzyme. So now this be the enzyme layer, okay, whereas this one going to be the enzyme layer. So the enzyme and enzyme. Now we got to know what is the enzyme. It's a outer layer of pollen grain. Okay. Now the enzyme is made up of cellulose and pectin. Enzyme is made up of cellulose and pectin. Okay. Where the enzyme is made up of one of the most hardest organic material called sporopollenin. Sporopollenin is a thing present in the enzyme. So now remember, enzyme makes the hardest coating here. It is not biodegradable. It protects the inner content of the pollen grain from the outer atmosphere. Now, looking into the options of the given statements, see, we got to know that the enzyme is the hardest outer covering. So, this statement is correct. It is made up of cellulose and pectin. This statement is wrong because the enzyme is made up of sporopollenin. And next, it withstand high temperature. Yes, the pollen grain withstand high temperature, means because of the enzyme. Enzyme withstands the high temperature and it won't get degraded by any actions of highly acidic content or the alkalis, and also it cannot be degraded by any action of known enzyme. So this statement is also correct and this one is also correct. So we got to know A, C, D are the correct statement, B is the wrong statement. So we can tell here option C going to be the correct answer. A, C and D are the correct statement.